Welcome to the lecture series in food microbiology. In this lecture, we are going to see the anti-nutrient and allergenic components which are present in the normal food components which we consume, be it the plant source or from the animal source. There are three broad classes of anti-nutrients: anti-proteins, anti-minerals and antivitamins. Antiproteins, these are substances that interfere with the digestion, the absorption or utilization of proteins. See, when we call anti-nutrient, anything, any component which can interfere with the bioavailability of what is essential, be it a protein, a mineral, a vitamin, iron, whatever, that has to be bioavailable. It has to be in a form which can be accepted by your body for its utilization. So, when any component interferes with the basic metabolic activity of absorption of the nutrition from the food, we call it as an anti-nutrient component. And these can be antiproteins, which will inhibit protein absorption, anti-minerals, which will either interfere with the absorption or interfere with the activity of the mineral components, and antivitamins. So, the first among those we are going to talk about antiproteins. So, these are substances that interfere with the digestion, the absorption or the utilization of proteins. And antiproteins occur in many plants and in some animals. Various protease inhibitors inhibit proteolytic enzymes in the gut, usually by binding to the enzyme's active site. See, proteins have to be broken down into their subunits and finally to the component amino acids for it to be absorbed by the body. So, for these we have proteolytic enzymes which are present. So, if the protease inhibitors bind to these enzymes active site, then those enzymes would no longer be able to go and bind to the proteins and break them down. So, that particular protein will not be broken down. Hence, your protein intake, even though you have had your normal protein intake, the absorption would be interfered with. Lectins are antiproteins that have binding sites for the cell receptors, similar to what antibodies have. Lectins, otherwise called as hemagglutinins, can agglutinate red blood cells. These are all examples of food which do contain some amount of lectins. Ovomucoids and ovoinhibitors, they are found in eggs and they inhibit the proteolytic enzymes. The egg whites contain chymotrypsin inhibitors. Many are sensitive to heat treatment and are inactivated at boiling temperatures for 15 minutes. Trypsin and chymotrypsin inhibitors are found in legumes, vegetables, milk, wheat and potatoes. So, trypsin and chymotrypsin inhibitors, which are very common, most of them are destroyed when properly cooked. The trypsin inhibitor found in milk is only destroyed after heating at 85 degrees centigrade for at least 1 hour, which means that it is quite stable. Soybean, which is another legume which is commonly consumed, has pancreatic enzyme inhibitors. And in rodents, they have been shown that feeding the rodents with soybean, constant diet of soybean, have been shown to reduce growth in the rodents. Elastase inhibitors have been isolated from soybeans, beans and potatoes. Lectins have also been isolated from the legumes, soybean, peanut, lima beans, kidney beans, fava beans, lentils and pea and potatoes, banana, mango and wheat germ, which means that most of the vegetable crops and the protein sources which we consume do contain some amount of lectins, which normally are destroyed by either the cooking or the processing methodologies, so that they do not become toxic or they do not become an anti-protein factor when we consume. And these plants do produce these, as these are trying to tell the human being, plants produce lectins in response to their being attacked by either a pathogen or a predator. So, that is a evolutionary response of the plants to produce lectins to protect themselves That is why that becomes toxic or a factor to the animals which consume them. These compounds bind avidly to the intestinal mucosal cells and interfere with the amino acids, thyroxine and fat absorption. Therefore, lectins are goitrogenic. So, in addition to being antiproteinaceous factors, they are also goitrogenic because they bind to the intestinal mucosal cells and they interfere with the thyroid hormone thyroxine. But there are some advantages of having a lectin free diet. That is, like, you have increased bioenergy, digestion is improved, you have that feeling that yes, you have the energy enough to do a lot of work, bloating is lessened, mental energy and mood swings are less, you have a high mental energy, and weight gain is controlled. Ricin is a toxic lectin which is isolated from castor bean, and it is notorious for causing death of children who eat raw castor beans. And ricin's mode of action is through intestinal cell necrosis. Most lectins are inactivated by moist heat, hence steaming is an effective processing technique used to inactivate ricin in castor oil. Antiminerals. Antiminerals are substances that interfere with the absorption or metabolic utilization of minerals. Some examples are the phytic acid, oxalic acid, glucosinolates, dietary fiber and gozipol. Phytic acid's negative effect on iron absorption has been known for decades. Phytic acid is normally present in the bran and germ of many plant seeds and in grains, legumes, nuts and species. In addition, phytic acid can also compromise the absorption of magnesium, zinc, copper and manganese. It usually forms precipitates like zinc phytate, copper phytate, etc. The formation of soybean protein phytate complex during processing has been associated with a reduction in the bioavailability of minerals like calcium, zinc, iron and magnesium. Fermentation and other food processing techniques have been useful in reducing the phytate levels. Oxalic acid, like phytic acid, reduces the availability of the bivalent cations. Sources of oxalate include rhubarb, spinach, beets, potatoes, tea, coffee and cocoa. Tree drinking was associated with concerns for calcium deficits via complexes, which apparently can be counterbalanced by using milk with tea drinking. Milk is a rich source of calcium and tea normally deficits calcium, so tea with milk can compensate for this calcium deficiency. Glucosinolates. These are goitrogenic and can inhibit iodine uptake into the thyroid. Rutabaga, turnips, cabbage, peaches and strawberries are good sources of glucosinolates. Dietary fiber, even without phytate, can affect the calcium, magnesium, zinc and phosphorus absorption. Collectively, the dietary fiber refers to the food components in plant cell walls that are normally not digested. Dietary fiber can act as ion exchangers and bind minerals. Gozipol. Gozipol is a phenolic compound isolated from the cotton plant, and it is capable of chelating iron and binding up amino acids. And this major toxic compound takes up to 1% of the seed's dry weight. Various processing techniques to isolate the protein can remove 90% of gozipol. So, this is actually the cotton plant. Ruminants normally can graze on the leaves, and this is a cotton seed which is a rich source of gozipol. So, this gozipol, when the ruminants consume them without the raw seeds, like processing can reduce 90% of the gozipol, and raw or unprocessed oil contains gozipol, which has genotoxic effects, and it has been proved in mammalian cell cultures, possibly through the production of superoxide ions and singlet oxygen induced strand breakage in DNA. All animals are susceptible to the activity of gozipol, but monogastrics, pre-ruminants, immature ruminants and poultry appear to be affected most frequently. Adult ruminants are able to detoxify the gazpacho by forming stable complexes with soluble proteins in the rumen, thus preventing absorption, something lacking in swine, pre-ruminants and young ruminants with only a partially functioning rumen. So, adult animals have a mechanism to escape from the toxicity of gazpacho by making stable complex. Thereby, this gazpacho will not be absorbed, whereas immature ruminants and swine which have partially functioning rumen, which is not fully functional as of yet, do not have this capability. Hence, for them, this is poisonous. Antivitamins. Substances that inactivate or destroy vitamins or inhibit the activity of a vitamin in metabolic reaction and increase an individual's need for vitamins are antivitamins. Ascorbic acid oxidase, an enzyme which is found in fruits and vegetables, can oxidize ascorbic acid. In addition to aerobic conditions, fresh juices lose 50% of the vitamin in less than 1 hour. So, when you are consuming fruit juice which is fresh, the moment it is made, it has to be drunk or consumed. On keeping, the vitamin quality will reduce because of the activity of the ascorbic acid oxidase. Raw fish contains thiaminase and this has antivitamin properties. And what it does is it splits the thiamine at the methylene linkage into 2 rings. Similarly, tannins found in plants also destroy thiamine. Likewise, mushrooms contain B6 antagonists and the linactine which is found in linseed oil is also an antipyridoxine factor. So, all these are antivitamin components which can interfere with the absorption, utilization and the functional activity of these vitamins. The next major disorder which we are going to talk about is a veno-occlusive disease. This veno-occlusive disease can be caused by 2 ways. It is actually with immunodeficiency. Along with immunodeficiency, this becomes a potentially life-threatening condition where the small veins in the liver are obstructed. There are 2 reasons for the manifestation of this disease, as I said. One reason is a high dose chemotherapy which is done before bone marrow transplant in the case of leukemia patients. The second factor which is related to food is excessive exposure to the liver toxic or the hepatotoxic pyrrolidazole alkaloids. So, one is chemotherapy which is given before leukemia, before bone transplantation. The second reason is the exposure to the pyrrolizidine alkaloids. The manifestation is weight gain due to fluid retention, increased liver size and the raised levels of bilirubin in the blood. Apart from chemotherapy, it can be caused by ingestion of pyrrolizidone alkaloids and these alkaloids are commonly present in high amount in some herbal teas. And this is also a part of a rare hereditary disease called as hepatic veno-occlusive disease with immunodeficiency because of mutations in the gene coding for a protein called as SP110, but this is very rare. The treatment for this disease generally includes supportive care, including pain management and possibly diuretics. In those with severe disease due to bone marrow transplant, difibrotide is the proposed treatment. The first report on veno-occlusive disease in 1920 was as a result of senecio poisoning in South Africa because of exposure to this alkaloid. Subsequent reports were mostly in Jamaicans who had consumed herbal teas. With the advent of bone marrow transplantation, subsequently whatever cases we have been hearing have been those undergoing treatment for leukemia. The next major topic under food microbiology which has to be discussed are the infections which are due to parasites, the intestinal parasites. Technically, all infectious agents including bacteria, fungi and viruses can belong to this category, but we have already discussed those diseases. But traditionally, the term has been reserved for an animal that derives its livelihood entirely from the larger animal or host, and under this category we talk about the roundworms, the hookworms, the tapeworms, the entamoeba, the giardia, etc. So, we include the protozoa and the worms, and we will exclude the arthropods in this particular category. All parasites are pathogenic, and the terms used to describe the diseases they produce end in the suffix -iasis like giardiasis or -osis like the toxoplasmosis. Some parasites are shed in the fecus and produce infection in those who ingest them with food or water. Other parasites are present only during a portion of their life cycle in animals used for food, and subsequently when these animals are consumed by humans, they will infect the human beings. In humans, they carry out the sexual reproduction and produce the eggs that will get shed along with the fecus. None of the parasites transmissible through the food or water is capable of multiplying outside the host. Protozoa. As you are aware, these are single cell animals. With respect to food or water sources, three types are important: Entamoeba histolytica, an amoeba; Giardia lamblia, a flagellate; and Toxoplasma gondii, a coccidian. Each has at least one active feeding form. This active feeding form is usually called as a trophozoite, and this can reproduce by simple division, binary fission usually, and a quiescent or a resting phase in which it retains its infectivity during the periods outside host. So, two types of, or I would say two forms are available. One is the trophozoite and a quiescent stage form. In the quiescent stage, it is still infective and it remains in the quiescent stage outside the host. Entamoeba histolytica. Transmission is by fecal to an oral route. The major source of infection is usually an individual who is asymptomatic but excretes the protozoa via fecus. Flies or roaches can be the vectors, and the organism can be transmitted during sexual practices. Infection is a worldwide problem, usually related to poor hygiene and sanitation. This is a picture of the Entamoeba histolytica. The habitat is the colon, but the protozoa can move to other organs in the body also. The active feeding form, the trophozoite, reproduces by binary fission, and this trophozoite can penetrate the mucosa, enter the bloodstream, and be transported to the other organs. The protozoa secrete enzymes capable of the cell membrane lysis. So, the host cell in which the organism enters gets ruptured. For Entamoeba histolytica, the human host is the most important one. However, these organisms have been found in dogs, cats, and other mammalia also. The cyst of Entamoeba histolytica is the infectious stage, and the protective cyst wall is resistant to many environmental conditions and is eventually excreted in the fecus. The clinical manifestation of Entamoeba histolytica can be grouped into four categories. The first are the asymptomatic carriers. These people contain the cyst, and the cyst gets excreted out via the stools, but the individual does not show any symptom or any manifestation of the illness at all. So, he does not know that he or she is infected. Non-dysenteric colitis. Here, the individual has non-specific symptoms like intermittent diarrhea, constipation, and abdominal pain, but you cannot say it full-fledged it is the disease. The cysts can be found in these people in the stool, and the trophozoites would be visible during the diarrhea. The third category is dysentery, people with dysentery with symptoms of bloody diarrhea, mucus, low-grade fever, and the colonic perforation with peritonitis. The fourth category would be amoebic liver abscesses, because the trophozoite has succeeded in perforating the liver and formed abscesses there. There is fever, there is pain in the upper right quadrant of the abdomen or in the lower right chest. The liver is enlarged, tender, and jaundiced. Metronidazole and chloroquine are the effective drug treatments for Entamoeba histolytica. This is normally, as I said, since the trophozoites are ejected out or excreted out with the stool, it is fecal-oral contamination which normally happens, and that is how a person will gain the infection. So, if you see, this is the macrocycle, you have the eggs assisting in the organism, The trophozoite inside the person, once it is ingested along, the trophozoite will migrate to the large intestine here, and then from there the trophozoite can move on or invade to the intestinal mucosa, and subsequently through the bloodstream it can infect sites like the liver, the brain and the lungs if the number of the parasites in the body goes beyond a particular number. It is an excessive invasion. If otherwise, the trophozoites which are present, which have been ingested, which have migrated to the large intestine, will divide or multiply by binary fission, then they will form a cyst, incestation. And this is your immature cyst, which is immature cyst. This is the quadrinucleate cyst, and these cysts are present in the stool, which are excreted out. And when this happens to be consumed by fecal-oral route by another person, the infection will pass on. So, this is the life cycle of the Entamoeba histolytica. The second parasite which we are concerned with is Giardia lamblia. This protozoa may gain access to the host by either ingestion of the cyst from the fecus or through sexual practices. This is a trophozoite, and the trophozoite here attaches to the mucosal surface of the small intestine, and these normally are confined to the lumen of the organism. The trophozoites reproduce by simple division, and since they adhere to the mucosal surface, they can interfere with the nutritive absorption of the host. Many animals have Giardia and they can infect the humans. Persons affected can be asymptomatic or show clinical symptoms of severe diarrhea. Both are possible, and if there is diarrhea, it can either be acute or chronic. The cysts of Giardia are relatively resistant to chlorination, but treatment with metronidazole is very effective. So, what happens is you have the contamination of food and water or even by fomites with the infected cysts. So, person ingests that. Inside the intestine, the trophozoid is there, the trophozoids are and they mature there. They attach to the mucosal membranes and interfere with the absorption in the host. But they do not, these trophozoids are passed out in the stool, but outside they do not survive in the environment. This is the cyst is formed. The cyst is the infective stage as well as the diagnostic stage. So, this cyst, when it is outside in the environment, forms the trophozoites. These trophozoites, as they mature, they again divide, the cycle continues, then they again form a cyst. So, this is how, and this cyst which is now present in the environment by contamination to the vegetables, fruits, water, fomites, everything, fecal-oral route again goes on. So, this is the life cycle of how a person can get infected with Giardia lamblia. Toxoplasma gondii. This organism, normally the host is cat, and many people have cats as pets. This is an intracellular parasite which multiplies in the intestinal mucosal cells of the host, which is the cat. And once it has infected the host, it can invade. So, it is initially in the intestine, but from the intestine it can invade other cells in other parts of the body of the cat, the host. So, the definite hosts are the members of the cat family. The oocysts, you can have congenital infection. The other animals can also form a secondary host. You have and once a pregnant animal is infested, it can become a congenital infection. Then the bradyzoids are formed. These are the secondary intermediate hosts. The bradyzoids are the ones which are formed like the trophozoids in Giardia. These are the bradyzoids. These infect human beings. If a pregnant female is infected, it will pass through the placenta to the child or the fetus, again showing a congenital infection. So, this is one source from the secondary host, or it can be from the pet cat. From the primary host, the oocysts can pass to the human beings. Entrance to the host is by ingestion of the oocysts from the cat fecus or by inadequately cooked meat, especially pork, lamb or beef. So, you clean up the litter and maybe not proper sanitation, and if the cat is infected, that is one route the host human can get infected. The second is you have feeding the meat, pork, lamb or beef, the animal which is a secondary host. The prevalence of Toxoplasma gondii in cats is dependent on it eating raw meat. It can infect the brain, the skeletal muscle and the retina. Common problems include ocular and neurological problems, jaundice, abortions or stillbirths, and malformation. Immunosuppressed individuals or patients with AIDS are particularly at risk from this organism. The usual treatment includes the use of pyrimethamine and sulfa drugs. However, pregnant women should avoid cats. So, if you see the life cycle, this is the main host, the cat. This is the unsporulated oocyst which is passed along with the fecus. This oocyst forms as a contamination in food, water or even in the soil. This is ingested by the intermediate host, which are any animal, swine, pork, lamb, cattle, anything. So, inside in the intermediate host, in the tissue, you see the cysts here, like the tissue containing the cyst. If this tissue is ingested by human being by consuming uncooked meat, the human being can get infected. The human can also get infected by contaminated food and water, and if it is a female, and she is pregnant, the oocyst can now pass on to contaminate or infect the fetus concerned. Summary. In this lecture, we saw how some food components can be anti-nutritive. So, rare amino acids, some anti-proteins or anti-lipid factors which can interfere with the absorption or sufficient utilization by the metabolism of the nutrient components of the body. We also saw parasitic infections that spread through contaminated food and water, and we learned how to take precautions to avoid them. In the case of anti-nutritive components, in many cases, proper food processing technology or proper cooking in many cases can inactivate those. So, their processing is sufficient to reduce the anti-nutrient components there. In the second case, sanitation and hygiene becomes more important, and the source of food as an even more important component in this part. The questions for this session are: describe the transmission of Entamoeba histolytica, Giardia lamblia and Toxoplasma gondii. As a food scientist, how will you eliminate the parasitic infestation of food? Thank you.